Hi, in this video, I would be talking about application of Galfor UA system in Drosophila. In my earlier video, I talked about how Galfor UA system works in Drosophila and what are the genetic crosses to perform this Galfor UA system in Drosophila. So now we would be talking about more about its use, application in research. So let's see step by step. So first thing we could do with Galfor UA system is we could check whether our Galfor driver is okay or contaminated. So here is an example. Here Actin 5C Galfor is crossed with UAS-GFP. So basically Actin 5C is under Actin 5C Galfor is under the Actin 5C promoter. So this Galfor is able to express whatever IUS construct is throughout the body. So this actin Galfor would drive the expression of GFP, which is a reporter here, in every portion of the body. So you can see there is a GFP in whole body of the adult fly as well as the whole body of the third instar larvae of these flies. And as every cell of the body has cytoskeleton, so every cell of the body is actually labeled by GFP. So let's talk about another driver. We can use ELAV. ELAV is a pan-neuronal driver. That means it's drive expression of UAS constructs in neurons. So when ELAV would express GFP, the GFP would be shown in the brain of the fly and other neuron spots. So here you can see in the adult fly, the area marked by yellow arrow is actually the brain. So it is actually fluorescing. So the brain is actually fluorescing. And also in the larvae, here is the brain, and brain is fluorescing intensively. And here are the segmental neurons, which are also labeled by GFP, as ELAV is a pan-neuronal driver. Similarly, if we cross a Galfor which is specific to eye, then we would find what? We would find the eye is also fluorescing. And this type of Galfor is GMR Galfor, which we would learn in a moment. Now if we cross GMR Galfor with UAS-GFP, we would find that the eye of the fly is actually glowing. So this means our Galfor line is not contaminated. And after doing this kind of experiment, if we don't find fluorescence in the eyes of the Drosophila, that means whatever Galfor line we have, it's contaminated with other flies. Now let's talk about some more application about Galfor UA system. So using Galfor UA system, we can use the eye of Drosophila as a genetic screen. So here one side we have a GMR Galfor line, and GMR is a eye specific driver, so it would drive the expression of UAS construct in the eye of Drosophila. So now in the UAS control we have a particular protein, say for XYZ protein, and now GMR would drive the expression of this UAS construct. Now, as a result, this protein would be overexpressed in the eye, and now we can see whether these overexpression causes any difference in the eyes, in the ommatidia of these eyes. So in this compound eye of the Drosophila, you can see there are a lot of ommatidia. So this is a control, and where there is no overexpression of any other proteins. And in the right hand side, in the panel B, you can see GMR is actually overexpressing protein XYZ. And in this particular case, on expression of these particular protein XYZ, there is a change in morphology of the eye. It appears that a portion of the eye is actually, the cells are actually getting dead. There is a cell death in the portion of the eye. So maybe these particular protein XYZ is actually useful for the development of photoreceptor neurons. We could primarily conclude that, and we can proceed to find out the answer. So this was another type of application that we can use. Another type of application is we can use Galfor UA system to do some localization experiments on proteins. So here we have a protein XYZ again, and we stain the protein XYZ with an antibody, and this antibody is rhodamin labeled. So, and you can see these proteins, this punctated structure in red, and the neuron here is actually, this is actually the neuromuscular junction I'm showing. So this neuron is actually stained with cysteine string proteins, the antibody against cysteine string proteins, which are actually presynaptic markers. So now when we merge these two pictures, we can see that this particular red protein is present in both muscle and neurons, but particularly in the neurons, this protein is co-localizing. That means this protein is present in neurons, but here the red arrow shows this protein is also present in the muscles. But as the yellow arrow shows, this protein is mainly present in neuron, because the concentration of the protein is huge in this neuromuscular junction part rather than the muscle part. So this type of inference could be drawn by this experiment. There are many more experiments of the Galfur-UAS system that we can perform. So these are few examples. If you like my video, give it a quick thumbs up, and please subscribe to my channel, and don't forget to leave your comments below. Thank you.